Hello, everyone. Thanks for joining us for CNEN's second annual virtual symposium, Advances in Drug Discovery and Development. I'm Matt Davenport, an associate editor with Chemical and Engineering News, and I'm delighted to introduce our next speaker, Emily Balskus, an associate professor in Harvard University's Department of Chemistry and Chemical Biology. Before joining the faculty at Harvard, Emily trained in a variety of fields, including synthetic organic chemistry, biochemistry, and microbiology. She tackled projects that covered asymmetric catalytic transformations and deciphered how photosynthetic bacteria produce small molecule sunscreens. Her research is now focused on problems she and her group find at the intersection of chemistry and microbiology. Their goal is to both improve our understanding of microbial biology and to learn how to better harness these organisms' ability to produce chemicals. If you'd like to learn more about Emily's work, check out our feature on her in our Talented 12 issue, and of course, stay tuned for her talk right now entitled Gut Reactions, Understanding and Manipulating Chemistry from the Human Microbiota. There will be a Q&A session immediately following the presentation, but you're welcome to submit your questions at any time. Just click the green Q&A button in the left corner of your screen. And if you need technical assistance during the presentation, please click on the support button located at the top right of your screen. Please note, CNEN does not endorse any company, product, or service that may be mentioned in this presentation. This talk will be archived and available on demand at This talk will be archived and available on demand at the conference website for 3 months after the live broadcast, until December 16th of this year. Lastly, if you haven't already done so, we'd love for you to join the Twitter conversation during today's event using the hashtag CENSIM15. And now I'm delighted to hand the session over to Emily.
Hi, everyone. Thank you for the introduction, Matt. I'm really excited to be able to participate in this virtual symposium. So I thought I'd start with an outline for today's talk. I'm going to begin with a brief introduction to the human microbiota and how we study it. Then I'm going to discuss my lab's efforts to better understand disease-associated chemistry from gut microbes. I'll then briefly comment on the implications of the human microbiota for therapeutics, and then finally wrap up with some concluding thoughts. So let's jump right in with an introduction to the human microbiota. So we live in a microbial world. Microbes, organisms like bacteria, archaea, and fungi, inhabit every conceivable place on this planet. And their chemistry, their metabolic activities influence numerous important processes really on a global scale. So these important organisms are what inspires the research in my lab. So the goal of our research is really to improve our understanding of the chemistry of microorganisms and microbial communities. We're really inspired by the fascinating biological roles of these organisms. We identify, study, and manipulate this chemistry. And in doing so, we gain knowledge that we hope will help us to better understand the important biological functions of microbes. So very broadly, we're interested in questions like these. How can we most rapidly uncover new microbial chemistry? How can we really zero in on the chemistry that's important for the function of microbial communities? And then finally, can we develop methods that will allow us to really manipulate important microbial functions in community settings, help us to understand their contributions to biology better? So I think that some of the most fascinating and important microbial communities we'd like to study are our own. So these are the trillions of organisms that live in and on us, the so-called human microbiota. So it turns out that we're actually more microbial than human. If you count up the number of cells associated with a person, you'll find that microbial cells outnumber our own cells about 5 to 1. And microbes inhabit many locations in and on the human body. But the most extensive community actually inhabits the human gut. And these are the particular microorganisms that the talk is going to focus on today. So microbes inhabit the entire length of the GI tract, a surface area that's about the size of a tennis court. They reach their highest density in the colon, which is actually one of the densest microbial habitats anywhere on the planet. So on average, we each have several hundred bacterial species in our gut, and collectively these organisms possess about 150 times as many genes as the human genome. So we've actually known for many decades that these organisms influence human health. They have many beneficial functions, so they impact important biological processes like the digestion of food, the development of the immune system. They provide protection against pathogens. There's also growing evidence that changing the gut microbiota can have negative consequences for human health. So altered gut communities have been associated with a growing number of human diseases, including IBD, obesity, and cancer. it's really important to recognize that we really don't yet have a good understanding of the mechanisms underlying this biological influence. So we have understanding these organisms are important, but we don't necessarily understand exactly how they're exerting this influence. So I think the question on this slide has really emerged as a major question in biology and medicine, something that's certainly important to address in the context of therapeutics. So what are the reasons for this knowledge gap? Well, a big reason for this knowledge gap actually has to do with the way we currently study the microbiota. So our information about gut microbes largely comes from DNA sequencing data. So we isolate DNA from a given body location, something like the human gut, and then we sequence it. Now there are various types of information you can get from DNA sequencing data. So one piece of information that's quite straightforward to get is information about phylogeny. This is because we have a good marker for bacterial phylogeny in sequencing data, the 16S rRNA gene. So this allows us to get information about who all the organisms in a community are and how they're related to one another. So whether they're present in a given body site like the human gut. We can also look at whether there are correlations between changes in community composition, which organisms are present, and health or disease states. So we've gotten a lot of, we've sort of observed a lot of interesting correlations doing this type of experiment. I'd say it's one of the reasons why there's a lot of excitement about the human microbiome right now, the really amazing correlations between changes in the communities and changes in biological phenotype. But this type of phylogenetic information really doesn't tell us anything about mechanism. Well, why not? The reason is that microbial phylogeny is very often not diagnostic of function. So a great example of this is actually to consider some organisms that hopefully we're all somewhat familiar with. So our favorite lab strain of E. coli, E. coli K12. And this strain of E. coli, 015787 or EHEC. So this is the pathogenic strain of E. coli that's associated with food poisoning. There's no doubt that these organisms are very different from a functional perspective. There's very different potential consequences for your health if you have one or the other of these organisms in your gut. Now, this difference in function is reflected in the whole genomes of the organism. So these two strains share only about 46% of their genome. But from the perspective of phylogeny, how the organisms are related to one another, their phylogenetic marker genes, they're almost identical. So the phylogenetic marker sequence that's associated with these organisms differ by only a single base pair. So if you sequence a microbiome and you look only at phylogenetic information, you can understand who organisms are, but that information doesn't necessarily tell you much about what organisms are actually doing. What we'd like to be able to do is to look at DNA sequencing data and get better information about biochemical functions, look at all the genes present in a given sample, and get information about what these organisms are doing rather than just who they are. And I think this is a really critical step we need to take if we want to begin to come up with hypotheses for how these organisms may be influencing biological processes. So my group thinks it's especially important to want to understand the metabolic functions of the gut microbiota, so these organisms' ability to make and modify small molecules. There's multiple ways in which the microbiota generates small molecules that are unique from the ones that our own cells produce. And I think this is a very amazing consequence of having a microbiota. We're actually exposed to a larger suite of molecules than just the ones that our own cells can make. This occurs to a really astonishing extent. So about 10% of the metabolites in human plasma are derived from microbial metabolism. And interestingly, metabolomics experiments in humans have found that in many cases, levels of microbial metabolites in human plasma can be very highly correlated with both health and disease states. So these are very interesting observations that suggest that microbial metabolites have the potential to be interacting with our own cells and potentially mediating biological processes. It turns out that we actually know very little about the genetic or biochemical basis for the production of the vast majority of these gut microbial metabolites. So to give you a sense of how little we actually understand, this slide shows some data from the Human Microbiome Project. So these are stool samples, genes that were annotated from stool samples from 148 healthy individuals. So we're getting a snapshot of the genes that are present in each individual's gut. So a given sample is shown on the x-axis, and a the y-axis are the percentage of different genes in each individual that can be annotated, that can be given a potential function. So we are actually able to annotate only a small fraction of the genes we find in these data sets. And even the functions that genes we can potentially annotate, the functions are quite broad and most likely inaccurate. So this is definitely a problem. Now we have the same issue for metabolites and metabolic functions. So on this slide I've shown some of the metabolites that we know are made by members of the gut microbiota and are the ones that have been shown to be most likely to be mediating important interactions with host cells. And we actually understand very little about the genetic basis for how these molecules are made by gut microbes. And even more broadly, we actually have not characterized the vast majority of molecules whose production is influenced by gut microbes. So the reference I've cited on the slide is a metabolomic study that was done in mice, and they were able to identify assign structures to only a small fraction of the plasma metabolites whose presence was impacted by having a gut microbiota. So we have a long way to go in our understanding of the chemistry of the microbiota. So my research group is seeking to address this problem. So we're using our understanding of chemistry, our ability to think about microbial metabolism and enzyme mechanism to try to elucidate the genetic basis for important gut microbial metabolic activities. So things like metabolites that are linked to disease. So we are applying this general approach to study many different types of gut microbial metabolic activities. And so today I'd like to tell you about just one of those projects. I'm going to tell you about how my group has discovered the genetic basis for a disease-associated microbial metabolic activity that produces this molecule, trimethylamine N-oxide, and our efforts to understand the contribution of this activity to human biology. So now I'll talk a little bit about work from our lab. And this project actually began in my group with an unexpected observation. And this observation actually was some data that came from a study run by Stan Hazen's lab at the Cleveland Clinic. And so he was interested in performing an untargeted metabolomics experiment aimed at understanding whether there were metabolites in human plasma that were predictors of future adverse cardiac events. So he ran an untargeted metabolomics study, and he found in this first group of 75 patients a correlation between adverse outcome and levels of this metabolite, trimethylamine N-oxide, in plasma. He went on to demonstrate causation in a mouse model, so you could feed an atherosclerosis-prone mouse trimethylamine N-oxide, and it would develop more severe plaques more rapidly. He then followed this study up with a much larger group of patients, following them for a time period of 3 years, and found the same correlation between plasma TMAO levels and risk for adverse cardiac events. So this is an intriguing correlation, but what does this have to do with the microbiota? Well, it turns out that trimethylamine N-oxide is ultimately the product of microbial metabolism. So it's derived from a gut microbial metabolite, trimethylamine. Trimethylamine is a volatile molecule. It's the molecule responsible for the smell of rotting fish. And in our bodies, trimethylamine is exclusively a microbial metabolite. So our own cells don't produce this molecule. TMA is produced by the activity of gut microbes, and one of the major sources of trimethylamine in the gut is actually the fermentation of the essential nutrient choline. So gut microbes are going to process choline anaerobically. They perform a carbon-nitrogen bond cleavage reaction to generate trimethylamine. And trimethylamine is further oxidized in the human liver by an enzyme called flavin monoxygenase 3 to give trimethylamine N-oxide. So this is an interspecies metabolic pathway. It has a gut microbial and a human component. And it turns out that both trimethylamine and trimethylamine N-oxide are connected to human disease. So increased plasma TMAO is associated with heart disease, as I mentioned. It's also associated with a growing list of other diseases. Now, the other metabolite, trimethylamine, has been involved, it's been appreciated for years, that it's involved in a different type of disorder, an inherited metabolic disorder called trimethylaminuria. So patients with this disorder have mutations in the gene encoding FMO3. This leaves them with an enzyme that's unable to convert all of the trimethylamine their gut microbes make to trimethylamine N-oxide. So they end up excreting trimethylamine. They have a very characteristic odor. So one of the things that's fascinating about trimethylamine is that this is, I think, a really rare example of a disease where a microbial metabolite, a gut microbial metabolite, plays a very well-defined role. It's also very interesting how these patients manage their disease. There's currently no drugs to treat trimethylamine. What patients do is regularly take antibiotics to tamp down their gut flora, and they really carefully modulate their diets. So presumably they're managing their disease by actually manipulating their own gut microbiomes. Okay, so because of both of these disease connections, my group became interested in which organisms in the human gut were actually responsible for fermenting choline to generate trimethylamine. And we kind of quickly realized that nothing was known about this. There was nothing known about the genetic or biochemical basis for choline fermentation. And this was a problem because you couldn't actually look at microbiome sequencing data and make any predictions about whether a given person's gut microbiome had the potential to do this chemistry or not. We were faced with a challenge, right? So given a disease-associated function that was linked with this complex gut community, how might we potentially identify the enzymes that were giving rise to this process? How might we connect this metabolic activity back to microbial genes? If we could do this, then we would have a way of looking for this activity in sequencing data and in gut microbial genomes. So we decided to take an approach to solve this problem. It's called genome mining. So we decided to try to use bioinformatics to rationally search for potential choline utilization genes in sequenced bacterial genomes, sequenced genomes of organisms that could carry out this metabolic activity. Now, in order to do this type of genome mining experiment, you need to have a very good biochemical hypothesis that guides your search. And so we generated a hypothesis by really thinking about the chemistry of choline fermentation. So it had been known for many years that choline fermentation involves an initial carbon-nitrogen bond cleaving event that generates two products, trimethylamine and acetaldehyde. So the acetaldehyde, it turns out, is what the organisms actually are using as a source of carbon and ultimately as a source of energy. Trimethylamine is simply released as a waste product. So we thought about the chemical logic being used here, the carbon-nitrogen bond cleavage reaction, generation of an aldehyde product, and we recognized a striking similarity to a different bacterial metabolic pathway, ethanolamine utilization. So this pathway also uses an initial carbon-nitrogen bond cleavage reaction. This reaction provides ammonia and acetaldehyde. And this is carried out by a well-studied enzyme, ethanolamine ammonia-lyase, which is an adenosylcobalamin-dependent enzyme. So this enzyme uses radical chemistry. And it makes sense that this enzyme should use radical chemistry, right? So this is a very difficult elimination reaction of a heteroatom. The C1 proton of ethanolamine is not acidic. You can't use acid-base chemistry to do this reaction. So nature's evolved the ability to use a radical enzyme. We thought about the potential parallel logic between these two pathways, and we wondered if the similarities might extend to the actual enzymes that were involved in these pathways. So specifically, we wondered if the enzymatic machinery involved in processing that key intermediate, acetaldehyde, might be shared between these two processes. So this hypothesis kind of illuminated a strategy for finding the choline utilization genes. We decided to look for homologs of the genes involved in ethanolamine metabolism in the genome of a choline-utilizing organism. So this strategy was successful, so we were able to identify a choline utilization or CUT gene cluster in this organism, Desulfovibrio desulfuricans. This is a sulfate-reducing bacteria that was actually isolated from the gut of the sheep. So in terms of what this cluster encodes, we did find, as we had hypothesized, homologs of the genes encoding the acetaldehyde modifying enzymes. We also see the genes shown in black there in the gene cluster, the structural components of something called a protein microcompartment. So this is a protein-based organelle that certain bacteria use to encapsulate metabolic pathways. There's a similar compartment used in ethanolamine metabolism, so it was not too surprising to find it in this pathway as well. And what really was a surprise was that we didn't see a homolog of the CN bond cleaving enzyme from ethanolamine metabolism. We didn't see a homolog of ethanolamine ammonia-lyase. Instead, what we saw was a different type of radical enzyme system, a system called a glycol radical enzyme. We named that enzyme choline trimethylamine-lyase, and we hypothesized that, again, due to the potential parallels in logic between the two pathways, that this radical-based enzyme might be the enzyme that was responsible for converting choline to trimethylamine and acetaldehyde. So we were able to use a couple different strategies to connect this gene cluster to choline utilization. So we had, in fact, identified the first pathway for choline fermentation in bacteria. So again, this discovery that the choline cleaving enzyme was potentially a glycol radical enzyme was pretty exciting from a couple of perspectives. So glycol radical enzymes are a large protein family found in anaerobic microorganisms. These enzymes use protein-based radicals in catalysis. So the active form of the enzyme is actually a radical that's situated on a glycine in the enzyme active site. And this radical species is very stable in the absence of oxygen. The radical has to be generated by the action of a separate activating protein, a member of the radical SAM enzyme family. All glycol radical enzymes are hypothesized to share certain mechanistic features. So in all the enzymes characterized to date, not only is there a conserved glycine, there's also a conserved essential cysteine residue in the enzyme active site. It's been proposed that it's actually this residue that mediates the reaction with substrate. So it's thought that the first step in catalysis is actually formation of a thiol radical. That thiol radical interacts with bound substrate through a CH abstraction. That generates a substrate-based radical, which can react or rearrange in various ways depending on the enzyme, giving you a product-based radical. That product-based radical can then react with that cysteine to regenerate the thiol radical and provide product. So the protein-based radicals in these enzymes are actually used catalytically. So glycol radical enzymes were known when we started our work to catalyze a pretty wide range of different transformations that are all important for primary metabolism in anaerobes. So they promote essential processes like ribonucleotide reduction. They play important roles in glucose metabolism. They also promote some more esoteric activities. This enzyme is involved in the anaerobic metabolism of toluene. The human gut is a largely anaerobic environment, so perhaps it's not too surprising that glycol radical enzymes turn out to be one of the most abundant and enriched protein families in the human gut microbiota relative to other microbial habitats. So we think that they're potentially a really important family to want to understand. So again, we had found this potential choline cleaving enzyme, and we thought it was a potentially new member of this enzyme family. And if that was true, it was kind of exciting from the perspective of chemistry, because that enzyme was catalyzing a carbon-nitrogen bond cleavage, which was not a type of activity that had been observed previously for this enzyme family. So we were able to test the hypothesis that this enzyme Cutc, or choline TMA-Lyase, was a new member of the glycol radical enzyme, basically using in vitro biochemical characterization. So we were able to heterologically express, purify, and examine the activity of both the activating protein, Cutd, and the glycol radical enzyme, Cutc. So this slide just summarizes how we think these enzymes are working based on a bunch of vitro biochemistry. So we think that this activating protein Cutd is responsible for generating the glycine-centered radical on Cutc. It uses a reduced 4-ion 4-sulfur cluster to homolytically cleave SAM, S-adenylyl methionine, generating methionine and a 5' deoxyadenylyl radical intermediate. That radical intermediate then abstracts a hydrogen atom from the active site glycine of Cutc, generating the active enzyme. And once this enzyme has been properly activated, then it can perform CN bond cleavage of choline. So this is what we observed when we reconstituted the enzymes in vitro. We've been able to completely reconstitute this process, we detect trimethylamine formation in assays only in the presence of all the assay components and under strictly anaerobic conditions. We also screened a number of other potential substrates, including other trimethylammonium-containing metabolites, things like carnitine and betaine. We also looked at other ethanolamine derivatives, and so far we've seen no reactivity towards anything but choline, since this enzyme appears to be quite specific. More recently, our efforts to further understand the mechanisms of this enzyme have focused largely on trying to understand what features of it actually differentiate it from other glycerol enzymes. And we've gained a lot of this information from some structural biology that we've done in collaboration with Kathy Drennan's lab at MIT. So recently, we were able, in collaboration, to solve the crystal structure of this enzyme. So Cutc, the tetramer in this structure, it displays the common fold that's characteristic of the glycerol enzyme family. And we were actually able to solve a bunch of crystal structures of this enzyme. The one I'm going to show you today has choline bound in the active site, buried here in the middle of the protein. And it's a 1.9 angstrom resolution structure. So if we zoom into that active site, you can see how choline is binding in the active site. So you can see choline here, and you can also see a single water molecule that's bound in the active site. So we can take a look at this structure, and we can get some information about various aspects of the reaction. So we can get some information about how the reaction is likely being initiated. Again, here's choline. Here is the glycine, the site of the glycyl radical. If you compare where this glycine is to where choline is bound in the active site, you see that it's actually quite distant. In between, perfectly situated, is this cysteine residue, and so this is consistent with the hypothesis that it's actually a thiol radical that's mediating chemistry with substrate. I also noticed that there is very short hydrogen bonding interaction between the C1 alcohol, choline, and this glutamate residue. And that's consistent with this glutamate being the general base that's important for catalysis, plays a role in forming acetaldehyde. So we've confirmed the importance of all these residues using site-directed mutagenesis. All these mutants possess no catalytic activity. And I'll just note that a similar set of residues is observed in another glycerol radical enzyme that does elimination chemistry, glycerol dehydratase, or GDH. One other thing I'll note is the dihedral angle we see about the C1, C2 substituents, and choline. This situates the pro-S hydrogen atom, which is the one we'd expect to be removed from C1 based on this structure, almost perfectly antiperiplanar to the departing trimethylammonium group, potentially suggesting that this is a direct elimination reaction. So we were also really interested in understanding how the protein scaffold might be interacting with the trimethylammonium group of choline. So we can take a look at some of the residues that might be involved in those interactions. So we have an aspartate residue here that is almost surely participating in an electrostatic interaction with the positively charged trimethylammonium group. We also see evidence for the involvement of multiple kind of hydrogen bonding-like interactions between the lone pairs of heteroatom-containing side chains, protein including this aspartate, and a couple of tyrosines, also the lone pairs of the water molecule here. So these are hydrogen bonding like interactions between those lone pairs and the CH bonds of the trimethylammonium methyl group. So these types of interactions are observed in other enzymes that make bind or consume trimethyl-lysine residues. So they're not new, but this is, to our knowledge, sort of the largest collection of them that's been seen in any enzyme. So to date, there's a lot of these interactions in this structure. And so we think potentially they may be important for helping to mediate the CN bond cleavage reaction. So we're in the process of using mutagenesis and enzyme kinetics to better understand the roles of these residues in choline binding and catalysis. We know already that several are very important for function. Okay. So our understanding of the mechanism of this reaction is still very much a work in progress. I wanted to share one potential mechanism you can draw that would be consistent with what we see in our crystal structure. So again, we think that the thiol radical is abstracting the pro-S hydrogen atom from C1 of choline. This would generate a substrate-based radical almost certainly you have a direct elimination of trimethylamine. It could proceed through an intermediate that's been deprotonated C1 alcohol. Once you release trimethylamine, you form an oxalo radical, and this could react with the thiolcysteine to give you the thiol radical back. We're in the process of doing some computational work, doing some additional experiments to probe this mechanism. But so far, this proposed mechanism is consistent with all the assays we've done so far with labeled substrates. Okay, so we're doing quite a bit of work to understand sort of this enzyme and molecular details underlying how it functions. Why is this important? Why do we care about how this enzyme is actually working? Well, it turns out this information, the combination of information about activity and information about structure, allows us to really understand the sequence-function relationship of this enzyme. And that, in turn, gives us information that actually helps us to differentiate this enzyme from other members of the glycoradical enzyme family. So there are lots of these things in microbial genomes, in sequencing data from the human gut. We really need to be able to differentiate this choline-cleaving enzyme from other members of the family if we want to really be able to accurately identify organisms that have this metabolic activity. Our understanding of the structure has allowed us to do that. We know that all of the residues that interact with the trimethylammonium group are very conserved among choline-cleaving glycoradical enzymes, but are actually unique to that set of GREs. They're not found in other members of the family. So we can take this knowledge, we can then go back and do further bioinformatics. So we can mine genome sequences, mine metagenomes, and for the first time, really accurately identify potential choline-degrading organisms. So we have found that the Cutc gene and the Cut gene cluster are widely distributed in the genomes of gut bacteria and also in human gut metagenomes. We also were certain when we did this initial analysis to actually get some of the strains we identified bioinformatically in our lab and make sure that the presence of the Cutc gene in an organism's genome is actually diagnostic of that organism's ability to degrade choline. So I'll share some of those results with you. So we've identified the Cut gene cluster in over 400 bacterial genomes. This data is a little over to date. We now see this cluster in more like about 600 bacterial genomes. The majority of these organisms are human-derived. We see the cluster in both pathogens as well as commensal gut microbes. And there's also a large number of environmental organisms and organisms that live in close association with plants and animals. So we were able to detect Cutc genes in almost every stool metagenome from the Human Microbiome Project. These are gut microbes from healthy people. So this gene is present in people. So that's very exciting. These genes are always associated with cut clusters and really display very high degree of sequence similarity, despite, as you'll see, the fact that they're distributed over a wide range of bacterial diversity. So this sort of shows you the distribution in different environments. What about the types of organisms that have this cluster? So we were initially kind of surprised to find that, this pathway is not limited to a small subset of microbial diversity. It's not limited to a single phylogenetic group of organisms. We find this pathway in organisms from multiple bacterial phyla. So we see it notably in Firmicutes, which are one of the two major phyla that dominate the human gut microbiota. And we also see it in a lot of proteobacteria. Now, this wide distribution suggests potentially that this pathway has been passed around in these organisms and acquired through horizontal gene transfer. We did a phylogenetic analysis of this protein sequence, and that analysis supports the hypothesis that this has been acquired through horizontal gene transfer. The other piece of evidence that supports that hypothesis is the uneven distribution of the cluster across individual groups of bacteria. So on this slide, we're looking at the different genera of cut cluster-containing organisms that are found in the human gut, and the percentage of the total genomes of that genre that actually have the cut cluster. And so you see a really wide range in terms of carriage of the cut cluster among these different phylogenetic groups. So it's not easy to look at an organism that's of a particular phylogenetic group and really make a prediction about whether that organism is likely to have the cluster or not. So this actually has important implications. So the phylogenetic composition of the gut, so just knowing which organisms are present in a gut community, is not likely to be a good predictor of this metabolic activity. So I think this is likely to be true of many other metabolic pathways as well, because of issues like horizontal gene transfer. So this is important, right? If we want to be studying particular metabolic pathways, particular functions in this environment, it's not enough to know which microbes are present in a community. We have to have methods of directly detecting our function of interest. So we're in the process of developing such tools that, things like selective PCR primers, things that will allow us to detect the abundance of the cut genes, transcripts, and proteins. We'd like to detect these genes, transcripts, and proteins in both model hosts. So we're doing that in collaboration with Federico Rey at the University of Wisconsin. And we also like to be able to look for the markers of this function in samples from human patients, and ultimately try to see if there really is a correlation between the abundance of this specific bacterial metabolic pathway and the levels of metabolites like TMAO and TMA in human plasma. Okay, so we're also working to identify small molecules that can inhibit choline metabolism, inhibit TMA production, and hopefully in that way manipulate levels of TMA and TMAO in model hosts. So we envision using such molecules as tool compounds, primarily to help us decipher the role of this metabolic activity in the gut microbiota, really see if this particular pathway has a connection to disease. One could potentially imagine using this strategy therapeutically, particularly for people who have that disorder trimethylamine. One other thing I'll just mention is that our studies of this enzyme, choline trimethylamine-Lyase, have also led us to become interested more broadly in other gut microbial glyceradical enzymes. So we knew from the enzymes that had been characterized when we began our work that this enzyme family really plays really critical roles in a wide variety of primary metabolic pathways in anaerobes. We also knew that these enzymes were widespread in the gut microbiota, and so we're currently working to better understand which enzymes are actually present in gut communities and predict what they might be doing. Now, in order to do this well, we really needed a way of being able to take a global look at the family. So not just everything that was known or had been characterized, but all the predicted members of this family. So we've done that using a strategy called network analysis. So what you're looking at on this slide is a sequence similarity network. It describes the relationship between roughly 27,000 different members of this protein family. So the way this network is organized, that a single node, like this one here, represents a set of sequences that all have 95% sequence identity. And two nodes will be connected by an edge if they have a pairwise alignment score of better than e to the minus 300. So this is a way, when you construct a network like this, you construct it in such a way that you group characterized enzymes that have known functions, together in isofunctional clades. And so this is actually the clade that contains the choline trimethylamine-Lyases. And so the way we've constructed this network, the other biochemically characterized enzymes clade pretty nicely as well. And everything that's shown here in gray is actually a clade we can't assign a function to. So there are actually many clades of unknown function. This view of the network then shows you the members of the family that are found in Human Microbiome Project reference genomes. You can see that both the known and unknown clades are found in these organisms. So we're currently working on identifying the functions of several of these uncharacterized clades, and we're using data from human patients to really guide that analysis. So we're figuring out what unknowns are really abundant in people who are healthy or who have disease, and really focus our efforts on deorphaning those enzymes. We think that these glyceradical enzymes are going to be really useful markers for many bacterial functions because of their critical roles in primary metabolism, potentially give us readouts of both beneficial as well as harmful pathways. And again, in the case of disease-associated functions, things like trimethylene formation, there's the possibility these enzymes could be relevant for therapeutics. Okay, so this brings us then to the discussion of the microbiota and therapeutics. And as you can imagine, the information we're really rapidly learning about the human microbiota, and really over the past decade especially, has rapidly opened up new questions and opportunities related to therapeutics. So I think that one of the major implications of understanding the microbiota has been really gaining a new appreciation for the potential effects that therapeutics people may be taking might be having on our microbes. So this is, of course, particularly worrisome when it comes to antibiotic use. We appreciate that antibiotic use inflicts collateral damage to the microbiota. Marty Blazer and others have proposed that the rise in certain autoimmune diseases that we've seen over the second half of the last century may actually be due to overuse of antibiotics, and they may be causing the disappearance of microorganisms that may be interacting with the immune system in beneficial ways. So along with the, of course, critical issue of antibiotic resistance, I think that worries like this are one of the reasons people are rethinking sort of how and when we use antibiotics. In addition to being affected by drugs we might take, the microbiota can also have an impact on drug activity. They can directly metabolize drugs, changing their activity. So this is a particular example my lab's been involved in studying. This is an example of a gut microbe inactivating, reducing a functional group on the cardiac drug digoxin, abolishing its effectiveness in certain patients. Gut microbes can also, in addition to directly inactivating drugs through their own metabolic activities, they can also produce molecules, things like paracresol, that have to be metabolized using the same enzymes that are involved in the metabolism of drugs we take. And so things like the levels of paracresol your gut microbes are making can influence the pharmacokinetics and efficacy of other drugs. And so this inter-individual variation in gut microbial metabolism could potentially explain some of the different responses the different individuals have to taking drugs. So certainly understanding this aspect of the microbiota has implications for both drug development as well as personalized medicine. Okay, so another interesting question of whether gut microbes and the metabolites they may be making are an interesting source of new therapeutics. So can we actually isolate molecules from gut microbes that possess interesting bioactivity? So we can. One example of a molecule that's been isolated from the gut microbe Bacteroides fragilis and has immunomodulatory activity is this molecule polysaccharide A. We also, of course, know that gut microbes have the potential to make complex small molecules. So we can look at the genomes of gut bacteria, we can look at metagenomes, and we can find biosynthetic gene clusters that appear to encode the potential to make more complex metabolites. So I think this is an exciting area of research currently. Because we've co-evolved with our microbes as we've evolved, it's certainly possible that molecules these organisms make have really evolved to interact with targets and host cells. And so it's entirely possible that we could find interesting therapeutics by trying to better understand microbiota-host interactions. And then finally, a major question that has emerged in this area is the question of whether it will be possible to treat or prevent human disease by manipulating the microbiota rather than manipulating our own cells. So of course, one of the major successes in this aspect of microbiota research has been the use of fecal transplantation to treat Clostridium difficile infection. So I'm showing here a figure from this New England Journal of Medicine paper that came out a few years ago. This compared fecal transplant to the standard of care vancomycin and found fecal transplant to be superior to antibiotic treatment. And so I think that certainly the success of fecal transplant is a demonstration that changing gut microbes can provide beneficial therapeutic outcomes, at least in the case of this particular infectious disease. I think we've yet to see if this strategy will be effective for other diseases. But what are some of the other ways we have of potentially manipulating the gut microbiota? So one strategy is to introduce isolated bacterial strains into the gut community that have beneficial properties, so probiotics. These could potentially be engineered, perhaps. That's an area of great interest. Another strategy, instead of administering live organisms, would be to simply manipulate the nutrients that gut microbes see in a way that promotes the growth of beneficial organisms. So understanding the role of diet, its impact on the microbiota, and potentially the idea of adding specific ingredients that promote the growth of certain microbes, things like prebiotics, is one strategy that is interesting to consider. But what about this idea that I mentioned earlier? So are gut microbes themselves an interesting target for small molecule therapeutics? What would really be the potential benefits of doing something like this? Well, one potential advantage is the level of precision you might be able to have. If you think about the ways we currently have of manipulating the microbiota with small molecules, we're really limited to very blunt tools. So things like broad spectrum antibiotics, these are going to inflict damage on a large chunk of gut microbes. So what we're envisioning doing is trying to potentially develop small molecules that target specific metabolic activities. And I think this is a really complementary approach. And such inhibitors, if we can develop them, could be really useful tools for deciphering the roles these metabolic pathways may be playing in complex microbial communities. There's not a lot of ways currently of manipulating activities in complex microbial communities with any sort of precision. And again, in the context of certain diseases, this could potentially be a therapeutic strategy to consider. Now, certainly there are many challenges associated with this approach. Many of these challenges are similar to the challenges associated with developing antibiotics, most notably the difficulty of accessing intracellular targets. The diversity of potential targets, certainly that's something we see with the choline metabolizing enzyme, that it's distributed across a wide range of bacterial diversity. So there's certainly challenges that will have to be faced in considering this approach to therapeutics. So it is interesting to note that this has been done successfully. So there's proof of concept for this idea out there. So this slide shows work from Matt Redenbaugh's group at UNC Chapel Hill. So he was able to actually use in vitro high throughput screening, so with an actual isolated enzyme assay, to identify an inhibitor of beta-glucuronidase enzymes from gut bacteria. So this is the molecule that he discovered. So this molecule was effective in preventing this reaction. So this is the removal of glucuronide from the cancer drug, irinotecan. This reaction, the bacteria remove this sugar in the gut to use the sugar as a source of carbon. This results in a reactivation of irinotecan in the gut lumen, and this is responsible for a problematic side effect of this cancer drug. What Matt's lab found is that administration of this molecule to mice actually prevented this dose-limiting side effect. There's proof of concept for this idea, and I think that this is a very exciting area for research in the future. Okay, and of course, I think that developing small molecule tools to probe this aspect of human biology is potentially a way in which chemists can make a really critical contribution to our understanding of the microbiota. Okay, so some conclusions, takeaways from this talk. So I think going back to my lab's own research, what we've accomplished is to go from a disease-linked metabolic activity, the fermentation of choline to form trimethylamine. This activity was associated with a complex microbial community. We've been able to go from that activity back to a specific enzyme whose role within the microbiome we're beginning to understand, and we can now think about beginning to target with small molecules. So our understanding of chemistry not only helped us to find this target, it also helped us to understand its distribution within the microbiome. And we think that generally, the infrastructure we've put in place, this approach, will hopefully provide us and others with a roadmap that could be used to study other disease-associated microbial functions. More generally, I hope I've really conveyed that an understanding of chemistry, the ability to think about how enzymes work and how metabolism works at a molecular level, can really play a key role in identifying new enzymes in the microbiota. This work has also certainly revealed some of the challenges associated with targeting pathways from gut microbes, namely the broad distribution of functions across a range of organisms. And finally, I hope I've convinced you that gut microbial metabolic activities and gut microbial metabolism is really a potentially exciting new frontier for drug discovery and therapeutics. So with that, I'll just quickly thank the people who've done this work. So most of the work I talked about today was the work of a single very talented graduate student, Miranda Bodea. She's been joined recently by two postdocs, Dr. Ana Martinez del Campo, who did all of the microbiology and bioinformatics with Cutc, and then also Spencer, who has done all of the more recent network analysis and bioinformatics with lysoradical enzymes. So we have a number of terrific collaborators as well, and I thank all the agencies on the slide for financial support. All right, so I guess it's over to Q&A.
Yeah, thank you so much for that fantastic talk. So we've got a lot of great questions to get to. So I'll start off with I guess a broader reaching question, which is you talked about at the end that there are a number of diseases that could tend to be treated with this strategy. Can you talk about some of the examples that you think are maybe most interesting right now?
That are most interesting to potentially treat? Well, I know that there's a lot of interest in examining IBD, so Crohn's disease and colitis, and there's definitely, I think, evidence for the shifts in the microbiota that are associated with those two diseases, really helping to drive the disease phenotype. It's still not clear whether the disease is triggered primarily by a change in the microbiota, but certainly once you have that dysbiosis phenotype, you promote a microbial community, or microbial community is established that's pro-inflammatory, and so I think that the understanding we have of the role the microbiome plays in IBD is creating much excitement about the possibility of being able to sort of use that knowledge for therapeutic development. So that's one example of a disease.
Excellent, thank you so much. So this next question is going back to the association of TMAO with cardiac disease. And the question is, do you think it's the TMAO itself that's the culprit, or is it just a marker of the trimethylamine? And I guess how can you differentiate between those two states?
That is a question everybody wonders about when it comes to TMAO and heart disease. So the primary piece of evidence for causation in TMAO actually directly playing a role in the progression of atherosclerosis came from the original 2011 Nature paper from Stan Hazen's lab, where he actually fed TMAO to mice, to the ApoE knockout mice, and saw enhanced accelerated plaque formation. So he could see that effect when he fed TMAO. He could also see that effect when he fed choline. But what was interesting was that the effect with choline was dependent on the mice having an intact microbiota. So if he first gave the mice antibiotics to knock down their gut microbes, feeding choline no longer had a adverse effect. So that was really kind of further evidence that it was really TMAO in that mouse model that was, having the direct impact on plaque formation. Now, of course, that's one mouse model. I think in humans it gets more complicated, and there are definitely several aspects to that correlation that give one pause in terms of thinking about whether the direct sort of causation for a role for TMAO actually makes sense. So one thing to consider is that, of course, one of the best dietary sources of TMAO is marine fish. There's no epidemiological risk for heart disease associated with eating marine fish. In fact, it's the opposite. Of course, it's complicated because when you consume marine fish, you consume much more than TMAO. But yeah, so there certainly is a question in humans if it is really more than just a correlation or not. You can imagine that if you have a disease like cardiovascular disease, you change as a host. The environment you provide for your gut microbes changes. And it's entirely possible that your body actually changing promotes the growth of organisms with slightly different metabolites. And so that could be why we see changes in certain metabolite levels. But yeah, so no one has yet to identify a mechanism, a target by which TMAO would actually modulate a pathway that would make sense in terms of heart disease. Work from Stan Hazen's lab and others has shown that when you kind of artificially manipulate the expression levels of FMO3, you see changes in cholesterol metabolism. And that's not something that you would have, anybody would have thought would be the case. And so there certainly are some hints that maybe there are there are differences in cholesterol metabolism when you have high levels of TMAO or high levels of the enzyme that's generating TMAO. But it's still very early days in our understanding of the host side of that correlation.
Great, thanks a lot. I know that there are actually a couple of great talks coming up at 3, so we'll wrap up with a few more questions here, so we get people over to that. So the one I have here right now is, could the increased TMAO or TMA plasma levels related to FMO3 absence or low activity polymorphs? I guess my understanding of that question would be, how do you, I guess, identify that what you found is in fact what's responsible for these elevated levels?
Repeat the question. Is it written down somewhere?
Question 12. Question 12.
Increased TMA. Could increased TMA plasma levels be related to FMO3 absence? Yes, and we would know that from the patients who have trimethylaminuria. So those patients have FMO3 dysfunction. So they have low activity. They're not able to convert all of the TMA that their microbes make to TMAO. So they do have increased TMA excretion in the urine relative to TMAO. And so it's actually an interesting group of patients to look at. A question I often get is are there any, is there any protection associated with having sort of a low activity FMO3? I'm certain that there are people studying trimethylaminuria patients now to try to answer that question. That mutation is not associated with any adverse health risks apart from sort of the psychological distress associated with having odor, the social debilitation that comes with that disease. Could they be protected against heart disease? Potentially, but we don't know the answer to that question yet.
Great, thanks again. Unfortunately, we are out of time. But again, thanks to Emily for that fascinating presentation, and thanks to all of you for tuning in and submitting such great questions. There are more talks for the rest of the day, starting at the top of the hour. You can find the full program on the agenda page located at the upper right-hand side of the screen. In the meantime, please step by the virtual exhibit hall and the lounge, check out our sponsors and exhibitors, or network with other attendees. Thanks again for attending. This is Matt Davenport for Chemical and Engineering News. Enjoy the rest of the symposium.